Scientists explain that the best way to understand any organism is to understand its genetic blueprint. So they say that they've learned how it functions, including which genetic traits make crabs particularly successful at...
For years, marine scientists had a grim nickname for certain parts of the Chesapeake Bay. They called them dead zones, not because the water looked unusual from the surface, but because of what was happening beneath it. In these areas, oxygen levels had dropped so low that most marine animals simply could not survive. Fish that swam into these zones would panic and flee toward healthier waters. Creatures that lived on the bottom, worms, clams, and crabs, often had nowhere to escape. Many of them suffocated. To researchers studying the bay, these areas were essentially underwater graveyards. So when a group of scientists proposed releasing 10,000 juvenile blue crabs directly into one of these polluted waters in 2012, the idea sounded almost reckless. The conditions that had already destroyed large parts of the crab population were still present. The water was murky, oxygen levels were dangerously low, and the sediment on the bottom was filled with decaying organic material. It looked like the worst possible place to release young animals. Yet the scientists behind the project believed something most people overlooked. Blue crabs were not just another species in the Chesapeake Bay. They were one of the key engines of the entire ecosystem.
And if that idea was correct, releasing those crabs might not just help the population recover. It might help restart the entire ecosystem. But before we get to what happened after those 10,000 crabs entered the water, it is important to understand how the Chesapeake Bay ended up in such a fragile state. And if you enjoy stories about science, nature, and surprising solutions like this one, consider subscribing and sharing this video. It really helps the channel reach more people who enjoy these kinds of stories. The Chesapeake Bay is enormous. It stretches over 200 miles long and covers roughly 4,500 square miles of water, making it the largest estuary in the United States. More than 150 rivers and streams flow into it, draining water from 6 different states, including New York, Pennsylvania, Maryland, Virginia, Delaware, and West Virginia. Everything that happens in those states eventually finds its way into the bay. For centuries, this system worked remarkably well. The bay supported huge populations of fish, oysters, and crabs. Early settlers described waters so full of oysters that ships sometimes had difficulty navigating through the reefs. At its peak, fishermen harvested tens of millions of bushels of oysters every year. Those oysters acted as natural water filters. A single oyster can filter dozens of gallons of water a day, removing algae and sediment as it feeds. But during the 20th century, that balance began to collapse. Industrial farming expanded rapidly across the watershed. Farmers began using large amounts of nitrogen and phosphorus fertilizers to increase crop yields. Cities grew, producing more wastewater and runoff. A large portion of those nutrients washed into rivers and eventually drained into the Chesapeake Bay. At first, the effects were subtle, but over time, the excess nutrients began fueling massive algae blooms across the bay's surface. When these algae blooms died, they sank to the bottom and began decomposing. Bacteria breaking down the organic matter consumed huge amounts of oxygen from the surrounding water. Slowly but steadily, oxygen levels dropped below the level needed for marine life to survive. This process created the dead zones scientists were now struggling to understand. By the early 2000s, these oxygen-starved areas appeared almost every summer. In some years, they stretched for miles across deeper sections of the bay. Fish abandoned these areas.
Shellfish populations collapsed, and many species that once dominated the ecosystem began disappearing. Among the hardest hit were the blue crabs. Blue crabs are more than just a regional delicacy. They are one of the most important animals in the Chesapeake Bay ecosystem. They are aggressive predators, but they are also scavengers. Blue crabs feed on worms, small fish, clams, and almost any organic material they encounter. In doing so, they help recycle nutrients and prevent excessive buildup of decaying matter on the seafloor. In a healthy ecosystem, blue crabs act like a natural cleanup crew. But as oxygen levels dropped and habitats deteriorated, blue crab populations declined sharply. In some areas, their numbers fell by more than 70%. And when those crabs disappeared, the ecosystem began to spiral downward. Without crabs consuming organic debris,
more decaying material accumulated on the bottom. That meant more decomposition and even less oxygen in the water. The bay had entered a dangerous feedback loop. By 2012, many scientists believed that certain areas of the Chesapeake Bay might remain permanently degraded unless something drastic changed. That was when Dr. Anson Hines of the Smithsonian Environmental Research Center proposed an unusual idea. Instead of waiting decades for pollution controls to slowly improve water quality, he asked a different question.
What if the missing species themselves could help repair the ecosystem? What if the bay didn't just need cleaner water? What if it needed its ecological workers back? The plan sounded simple, but it carried enormous risk. Scientists would raise thousands of blue crabs in hatcheries until they reached a more resilient juvenile stage. These crabs would be larger and stronger than newly hatched larvae, giving them a better chance of survival. Then they would release them into a tributary of the Chesapeake Bay called the Lafayette River, an area that had suffered severe environmental decline. If the crabs survived long enough to reproduce, they might restart the natural population cycle. But if they failed, the entire experiment would collapse. On a humid morning in June 2012, boats carried crates filled with 10,000 young blue crabs out onto the Lafayette River. One by one, the crabs were released into the dark water below. Then the waiting began. For several days, the scientists had no idea whether the animals had survived. 3 days later, they set out crab traps to check the results. When the first trap was pulled from the water, the researchers immediately noticed movement inside the cage. Blue claws snapped against the wire mesh as the crabs scrambled around. The team quickly checked the identification tags. These were the hatchery crabs. They had survived. That moment alone was encouraging, but survival for a few days was only the first test. The real question was whether the crabs could survive long enough to begin influencing the ecosystem itself. And that is exactly what began happening over the following months. As the crabs spread across the riverbed, they began feeding aggressively. They consumed dead fish, decaying plants, and organic sludge that had been accumulating for years. In effect, they were removing the very material that fueled oxygen-depleting decomposition. At the same time, their constant movement across the riverbed stirred up sediment and allowed oxygen to penetrate deeper layers of the soil. Slowly, sensors placed in the river began detecting subtle improvements. Dissolved oxygen levels near the bottom increased, organic waste levels dropped, and then something happened that surprised even the scientists running the experiment. Baby crabs began appearing in survey nets. The released crabs had successfully reproduced in the wild. That meant the population was no longer dependent on hatchery releases. The ecosystem was beginning to rebuild its own crab population. And as the crab population expanded, other changes began spreading through the food web. If you're enjoying the story so far, consider subscribing to the channel. Stories like this reveal how complex and sometimes surprising nature can be. The growing crab population triggered what ecologists call a trophic cascade. Small fish that had previously dominated the riverbed suddenly faced a new predator. Their behavior changed, allowing populations of clams and other shellfish to recover. Clams, in turn, filtered large volumes of water, improving water clarity. Clearer water allowed sunlight to reach underwater grass beds, enabling them to grow again. Those grasses provided habitat for young fish and crabs, strengthening the entire ecosystem. Within a year, the Lafayette River began showing measurable signs of recovery. Grass coverage increased, fish populations expanded, and the water itself became clearer. The success of the experiment eventually led to expanded restoration programs across the Chesapeake Bay. In the years that followed, hundreds of thousands of hatchery-raised crabs were released into other struggling areas. The results were consistent. When key species returned, ecosystems often began repairing themselves faster than expected. But scientists are careful to point out that the Chesapeake Bay is not fully restored. Pollution still enters the watershed from agriculture and urban runoff. Dead zones still appear in some areas during certain seasons. What the crab experiment demonstrated was not a miracle cure. Instead, it revealed something equally important.
Ecosystems have powerful recovery mechanisms, but only if the critical species that hold them together are still present.